going to begin the chapter of immune system and the lymphatic system. First, we need to have a basic understanding, right, of the cells that are part of the immune system. And these cells are called white blood cells. And the abbreviation you will see, right, WBC. And the components of white blood cells are as follows. We have neutrophils, eosinophils, basophils, lymphocytes, and monocytes. So I'm going to spend some time to go in depth about all of these cells. We're going to talk extensively about all of these cells today, how they play a role in the immune response, right? So you're going to be all experts by the end of this lecture in terms of how they exert their effects, right? We see that they make up, if we take your blood, right, sample, they make less than 0.1%. And that's normal. Why? Because for the most part, right, when these cells become more excessive, I should say, the production of these cells increases. At what conditions?
Infection.
Right, infection or some type of viral infection, acute infection, could be chronic infection, right? But for the most part, right, we don't have acute elevations of these white blood cells under normal circumstances. So before I get into all these slides, right, I will basically give you some schematics in terms of you know, the classifications, what they do, the numbers, and all of that. And then I think we can go through all the slides slowly. Right? So overall, right, all of them are known as white blood cells, as we just talked about. And then they're further subdivided into granulocytes. And they have, sometimes they called agranulocytes or non-granulocytes. So what is this granules, right? So the granules are inherent to the specific cells we're talking about, right? So the granulocytes specifically that I am referring to is the neutrophils, the eosinophils, and then we have the basophils. And then the non-granular sites, or agranulocytes, sometimes they refer to, is the monocytes. And then we have the lymphocytes. Right? This is the same thing we just saw on the picture of the previous slide. Now, the way they differentiate them, or the way they look at them, is basically something's called as, right, cytology or histology. They basically take these cells and they put them into the microscope, and then they look at all these granules, right? And we're going to talk about, right, all of them, specifically the differences. So when you look at the microscope, what you're going to see in the neutrophils, right, they're usually multilobated. So they have their nucleus have multiple lobes, right? So you'll see 2, 3, 4 or more. So that's the appearance under the microscope. So they have multilobated nucleus, right, is usually the hallmark sign of a neutrophil. Eosinophils, it also has a nuclei, but it usually it's usually 2, right? So it's bilobe nucleus, and usually it may look like spectacles, like glasses, right? So it's bilobe nucleus, or there is 2 nuclei there. For basophils, it's usually, it looks like an S-shaped, and it's also bilobed. This is the type of appearance it will have, S-shaped or bilobed. In monocytes, it usually looks like a kidney. So the nuclear material is like kidney-shaped. And then in the lymphocytes, the nucleus takes up pretty much the entire cell. right? So this entire content is the nuclear material or the nucleus. Now, we were talking about these granulocytes and granulocytes. So let me ask you this question. So all of them need to have some sort of granules, right? Which I haven't explained to you what they are. And these, based on the name, they say non-granulocytes, so we assume, right, no granules are present. So do you think these cells have granules and these do not? What do you guys think?
Yeah.
Crystal, you want to answer?
Oh, I'm going to go with whoever said yeah.
You're going to say yeah. So in the yeah, by yeah, you mean what? These have the granulocytes
They do have granules, and then the
non-granulocytes do not have granules, right? So
yeah.
in a way, they kind of made this a little confusing, and I agree with that. So truly speaking, all of these cells have granules. However, these have specific granules. But the non-granulocytes also have granules. However, they're called non-specific. So when we start to differentiate these cells, and when they were finding these cells on slides, when they were taking blood sample from patients, initially they were plating them, right, with cytology, looking under the microscope. So what they saw with neutrophils, they had two types of granules. They had ones that took on a blue color, and then they had ones that took on the red color. So the ones that take on the red color, we call them acidic color, right? Or eosinophilic. So red color loving is eosinophils. Red color is also acidic color. The blue color, B for blue, B for basic. So blue color is basic dye, and that's basophilic dye. So neutrophils, they found, contain both the red and the blue granules, right? Eosinophils, when they plated them, right, because they took on the red color or the acidic color, they call it eosinophils, right, or red color lovers. So this was the acidic color. And then the basophils, they took on the blue color, and the way you want to remember is B for blue or B for basic. Right, so this is how they were able to differentiate them under the microscope for different patients. The monocytes and lymphocytes, they have non-specific granules, or sometimes this non-specific will be also referred to as lysosomes. And you will see some granules, right? I'm not going to put any dyes on them, but they do have granules. They're usually non-specific. Another names would be lysosomes. Another name would be azurophilic granules, right? Or the primary granules. They all mean the same thing. But these cells definitely have granules. It's just that they do not take on any particular color. Now, when we take your white blood cells, right? And we put it in a test tube, right? We take it and we put it in a test tube. And we measure how much cells there is per 1 ml of blood, right? How much do you think on a person who is not having an infection, what is their total white blood cell count? Would anyone tell me how much white blood cells are found in 1 ml of blood for a normal person? Would you say?
Around 55,000.
55,000, I think that's a little too much. I think that person may be having an infection.
2,000?
2,000 is a little too low, right? They may also be having an infection. So the normal count is about 4,000 to 11,000, right? And that's about per cubic millimeter or per 1 ml, right, of blood. So if you're within this range, right, you have a normal amount of white blood cells in your body. If you're below this number, you may have a pathology. If you're above this number, you may have a pathology, right? So the normal number being 4,000 to 11,000. In the hospital, right? Sometimes they call it, they could call it DLC, right? Or let me say TLC, I should say TLC, total leukocyte count. So total leukocyte count basically means total amount of white blood cells that are found there, which this would be the normal number, 4,000 to 11,000. Now, there's another term that I kind of wrote it here, but you should be familiar with. It's DLC. So DLC stands for differential, right, count. So this stands for differential leukocyte count. So the moment you start to talk about DLC, differential leukocyte count, you can no longer refer to the numbers, you are referring to percentages, right? So TLC is the total amount of white blood cells, right? And this was the number here, right? 4,000 to 11,000, we said. Right? But the moment you start talking about DLC, differential leukocyte count, we're getting into the percentage range. And why do you think we care about that? Why is it important to know the percentages of this differential leukocyte count?
Because each cell has a different job.
Correct. So Ian is 100% correct, right? Every white blood cell has a different job. So we're going to talk about specifically what their job is. Another terms, I would say it's because you may hear it, in the hospital, a doctor may order CBC with Diff, with differential, with Diff. That basically means they want complete blood count with this, with differential leukocyte count. And usually when patients come in and they suspect some sort of an infection, right? Chronic infection, acute infection, right? Or viral infection, they want this number, they want to see which cells are increasing so they can essentially establish appropriate diagnosis. So total leukocyte count, we said this, the total number of white blood cells, but differential leukocyte count is going to look at specific percentages of all these, and we're going to talk about that. Another term that I want you to be familiar with, right? The moment this number gets below 4,000, we call this condition leukopenia. The word penia usually refers to poverty. So when this number drops below 4,000, that usually is called leukopenia. And then the moment this exceeds 11,000, we call it leukocytosis. So you're making more white cells, right? So leukocytosis and leukopenia. So these two terms are also may be used in the clinical setting, so you're familiar, right? Now, let's talk about normal percentages. So what's normal DLC, right? Differential leukocyte count. So when you look at neutrophils, neutrophils are about 40 to 75% of total white blood cells. So they are the predominant white blood cell. Now, the numbers that I'm giving you, they may change from laboratory to laboratory, from book to book, but roughly they'll be in that range, right? So if you, let's say you're in your hospital and you want to see the blood work, you could, ask the nurse to, print you that particular page, right, of CBC with differential. So you could look. They will slightly differ, but those numbers will not be significantly different. So if you understand these numbers that I'm showing you here, you'll be in the correct ballpark, right? So neutrophil 40 to 75%. Now, if you're below this number, right, this is called neutropenia. Right, so you're below 40%, let's say you're in, 20%, you're going to be neutropenia. What do you guys suppose if it's above 75? What do you think it's called?
Neutrocytes?
What is it? What'd you say, Lindsay?
Oh, sorry, neutrocytosis?
So yeah, that would be a good guess, but medical science is not so easy, right? So they actually call it neutrophilia. So the higher amount of neutrophils in your blood is actually called neutrophilia. Now, this white blood cell, the neutrophils, right, multilobated, that's how we know it, multiple nuclei, has two types of stains, eosinophilic stain, basophilic stain. This is the predominant white blood cell when you have any type of acute infection, especially any infection that's pus producing. So you may have, for example, streptococcus, staphylococcus, right, infection, and which produces, pus. Neutrophil would be the first white blood cells that will rush to the site of that bacteria, right? And this has the capability of phagocytosis. So basically, it has the capability of trying to engulf the bacteria, trying to essentially, eat it up, and then break it apart with its granules, right? Lysosome granules to basically kill the bacteria. So anytime we see any type of acute infection, which is, we call it pyogenic infection, pus-producing infection, secondary to streptococcus, staphylococcus, or such bacteria, we see high elevations of neutrophils. And that condition we're going to call neutrophilia. The next one that we're going to talk about is essentially eosinophils. And the normal number is roughly 1 to 6% in your blood. That's if you do not have any type of problems. When do we see high elevations of this, right? We see high elevations of this when a person suffers an allergic reaction, or a person suffers parasitic infection, parasite. Right, like worms, any type of worms, helminths, right? Any type of parasites that colonize in your body, you'll see elevations in eosinophils, and also for allergic reaction. Now, when it's below 1%, it's gonna call eosinopenia, but when it's above that, right, we're still gonna keep the same term, right? We're gonna call it eosinophilia. So eosinophilia, the elevation of these, right, is seen especially in allergic reaction. We see in a parasitic infection, in certain neoplasms, cancers, you may see that elevation. You also will see it with certain collagen disease states. But for the most part, what you want to remember, in the allergic reaction and parasitic infection, this is the one that's get elevated. Now, basophils is the cell that's has the lowest numbers out of all of them. The numbers is 0 to 2%. You really have to search to find these cells on the slides. Sometimes you can't even find a single one. You have to go slide to slide and really try to find them, right? So basophils, elevation of these right above the 2% is going to be called basophilia. And then where do you see elevations of these? So you see these also in allergic reaction, especially type 1 hypersensitivity reaction, which we're going to talk about extensively today. So at these levels, right, this will be elevated. We see this, as I said, right, type 1 hypersensitivity reaction, which we will delve into much deeper. And the nucleus here, right, is usually S-shaped nucleus, where other conditions this can get elevated. In certain anemias, right, like thalassemia, iron deficiency anemia, you will see elevations in basophils, and conditions of that nature. Patients who consume lead, right, as lead poisoning, may also have elevations in basophils. But for the purposes, the ones you want to remember is you want to remember elevation in allergic reaction and type 1 hypersensitivity reaction. You will have higher abundance of these cells. Now, monocytes, we're going to go to non-granulocytes, but as we said, right, they still have non-specific granules or lysosomes. The TLC count for them is about 2 to 10% for your normal person. And usually, when do you see elevations of these, right? Usually you see elevations of these in acute infections, right? And you could also see them in chronic infections. Chronic infections like tuberculosis, for example. It will be an example of chronic infection, right? Like TB. And acute infection could be like, pyogenic, right? Streptococcus, staphylococcus reaction. Now, here is this again, right? As I said, this is a little different in terms of how you name this. So what do you suppose you call when you have below 2% of monocytes? Ian, you want to say it?
Monopenia?
What would you say?
Monopenia?
Yeah.
Monocytopenia?
Monocytopenia. So basically means cyto means cells and penia means poverty. And what would be when it's above 10%?
Monocytophilia.
Yeah. So here it's actually cytosis, right?
Oh, true.
Here's what becomes tricky, right? So remember, for your non-granulocytes, the terminology changes, right? In terms of when it's elevation. So when you have non-granulocytes, elevation is monocytosis, as opposed to all your granulocytes were neutrophilia, eosinophilia, basophilia, right? So this is what you want to keep in mind, right? And there's no stains here, so there's no blue color, there's no red color for the stains we just observed in these conditions. The next one is lymphocytes, right? So these cells, roughly speaking, depending on the lab, right, they'll be about 15 to 40% in terms of the amount you have. If you have greater than 40%, right, the term will be lymphocytosis, right? Same thing like monocytosis we saw here. Where do you see these elevations occurring? So these elevations usually occurring in viral infections and chronic bacterial infections. So another important thing I want to say, right? You notice how you have a crossover here, right? You see monocytes, they could be elevated in acute infections, they could be elevated in chronic infections. So the hallmark to know when you're dealing with which, if you have elevations of monocytes, which is monocytosis, along with neutrophilia, that's usually an acute reaction, right? So acute reaction of neutrophilia with acute elevation of monocytosis, you're dealing with an acute infection. If you have elevations of lymphocytes, right, lymphocytosis with monocytosis, you're probably dealing with a chronic infection. So those are the hallmarks to keep in mind, right? What else is important to tell about these cells, right? So we just said this, the percentages here that we saw, this was your differential leukocyte count, right? And in the hospital, sometimes the doctors will come in and they'll say, okay, nurse, I would like you to draw, CBCs with Diff. So Diff, they basically mean a differential count, right? And the reason why they want to know is that they want to know these percentages. And they want to look when the person is having, let's say, fever, or they have complaints of malaise, right? They want to see which of these white blood cells are elevated, right? And specifically the percentages of them, right? They want to know specific percentages. And the lab work that you get, the lab itself has their own values, but they will show you, right? They usually make those white cells that are elevated, they'll put it in bold color, and they will say, like, let's say normal value, let's say neutrophils is 40 to maybe 75%, and here it's 86%, and they'll show that the percentage is elevated. And what happens in that condition is basically his body is making more of those cells to send and fight the acute bacteria, right? The acute infection process. So this is how they are able, the physicians are able to put the appropriate diagnosis. Is patient comes in with chief complaint, they'll do their physical exam, and then they will order specific blood work they'll look at these numbers, and then they will be able to better state their diagnosis. The other important thing that I want to say, right? Monocytes, as long as they're in circulation, right? They're called monocytes. But the moment they leave the circulation and they go to the tissues, right? The moment they go to the tissues, they're no longer called monocytes. So in the tissues, and you want to know what they're called in the tissues?
T cells.
What is it?
T cells.
Not E cells. There's a more
T cells.
What cells?
T.
Oh, T cells. T cells are part of your lymphocytes, right? So I'm gonna spend some time to break them down even further. But the moment monocytes go into the tissues, they're called macrophages, right? You probably heard the term macrophages, right? They're the big eaters, right? Phage means to basically eat or engulf. So macrophages, these are your big eaters, right? And these cells, right? All these cells, neutrophils, eosinophils, basophils, they're also able to phagocytize bacteria. But these are the big eaters, right? These are the big eaters of your immune system. So any questions in terms of so far what we talked about, right? The total leukocyte count, differential count, these cells and their function in the body. We're gonna spend some more time talking about a little bit more in depth about these cells. But any questions thus far? Right. So if there's no questions, we're gonna proceed, right? So what else is important to know, right? So the other important thing that you want to know is when these cells are being made in your body, they're made in the bone marrow, right? So let's say, let me draw your circulation here, right? So we're going to say this is your circulation. And these cells, right, the white blood cells, specifically, I'm going to be referring to all these three, the granulocytes, the neutrophils, eosinophils, and basophils. They basically go through the same cycles when they're made in the body. So they usually, from the stem cells in your bone marrow, as they're made, they go through stages of maturation, right? As they mature, they start to make more specific follicle of these granules, so the cells can be differentiated, right? But so they go through these stages up until the final portion, right? When they're finally in the blood. And let's say this was your neutrophil. So we're going to say this is your neutrophil. And it has the multilobated, right, nucleus. It takes the basophilic stains. It will take the eosinophilic stains. And normally, once this comes to the blood, right, the lifespan of them in the blood is not so long, right? In the blood, they may live for several hours before, they get degraded, right, and they get broken down. And we're going to say this here is your bone marrow, right? So this is your bone marrow, and as your bone marrow is making the cells. By the way, where in your body we find this bone marrow? Where are these bone marrows found?
In the long bone.
The long bones, right? Essentially in your axial skeletal, right? So basically in your ribs, your long bones, right? Especially the top and the bottom of the bones. You also see them, right? The bones of the skull can make these. Basically, as the infant is born, all of their bones are making, right? Are able to make, right? All of their bones have the red bone marrow. But the moment we mature and we grow, right, it basically shrinks to, right, just your axial skeleton. So it's going to be your long bones, right, ribs, right, pelvic bones, skull bones, and this is where the maturation occurs, and they go through, different stages. But what's important to know, your bone marrow pool. So we're going to say this is your bone marrow pool of of these cells. They have about 5 times more, right, of these white cells as opposed to the circulation. So circulation has 5 times less of these cells circulating. And as these cells, divide, they have different names. It's not so much for you to remember all the stages. What's good to remember is that before they become neutrophils, right, this stage is they're called stab cells. Stab cells, or they are called sometimes band cells. So whenever they hear, right, there is increased, they'll call it like elevated bands, right? Elevated bands, or they'll say bands about a certain percentage, let's say greater than 10%. What that means is that whatever the person is, the infections they're having, is that their body, right, their bone marrow pool is now starting to dump the stores of these white blood cells, specifically neutrophils, basophils, eosinophils, into the circulation. So then we see these immature cells, these stab cells or the band cells, they are now moving, right, from the bone marrow pool, they're moving towards the circulation. So that's why we see elevation of band cells, which were essentially immature, right, or not fully mature neutrophils. And we see the elevations. These neutrophils that are in the blood, they could adhere to the vessel walls, right? Those that are adherent, we call them basically marginated pool. And those that are freely circulating in your blood, we call them circulating pool. So the moment the bone marrow pool dumps these into the circulation, we basically have elevated circulating pool, right, of these specific white blood cells, depending on what they dump, right? So neutrophil being the first white blood cell that's released for the site of injury or infection, that's what you may see. So you'll see neutrophilia, you'll see eosinophilia, you'll see basophilia in those specific reactions, right? So that's what we encounter. And we can measure, right, when we do CBC with differential, we could measure these band cells or stab cells, which are immature cells in this condition, right? So that is important to know about that. Any questions about that? Right. So no questions about that. The other thing I'm going to talk about is now let's get to the monocytes, right? And I specifically talked about when they leave the circulation and they go to the tissues. They have different names. They're called macrophages, right? I put this here. But there's even more specific terms. In the tissues, right, they could live for several days or even years at certain tissues. So what are what are they called? So let's say, this was your circulation, right? And we're going to say this was your macrophage. It had a kidney-shaped nucleus, right? And the moment it leaves the circulation, it goes into your tissues. So here I'm going to draw some tissues, right? So one tissue it can go is your central nervous system. It can go to your brain. Let me draw, right? Anyone remember from the neuro chapter what those cells are called? The white cells that are found in your CNS? Especially these macrophages. Anyone recall? So remember we talked about called microglia cells. So microglia cells is essentially the macrophages that are found in your central nervous system, right? Then in your lungs, we talked about, right, in the respiratory chapter, what are the names of the macrophages found in your lungs? They basically, these cells circulating in those tissues, and they're constantly surveying the environment. They're looking for the bacteria to basically phagocytize. Everyone recall what is the macrophages in your lungs called?
Lymphocyte.
What is it?
T lymphocyte.
So they're not they're not lymphocytes. Lymphocytes are different cells, right? So they're still macrophages, but the name in the lungs, they're going to be called alveolar. So they're called alveolar macrophages. So the reason why I bring these out is because the moment this monocyte leaves the circulation, right? They have different names. They're going to be called macrophages. In the central nervous system, they're called microglia. And the lungs, they're called alveolar macrophages. Their job is essentially to like patrol and look for foreign invaders. And the moment they see these foreign invaders, we're going to talk about some of the functions they're going to perform, right? One function will be phagocytosis, where they engulf the bacteria. Another function, they're also called, antigen presenting cells. So they can take that antigen, right? And then, right, Helmi, as you were talking about, then they could present that antigen to your lymphocytes, the T cells, right, and the B cells. Some other, tissues that you guys should be familiar with, right? You have the liver. In the liver, they call the Kupffer cells, right? They also found in your, in your bones, right? They're called osteoclasts. So the macrophage is also found there. What else? They could be found in your skin. They could be found in your kidneys, right? And another term for these cells, they're also called histiocytes. Right? So when they talk about histiocytes, they could also be referring to the macrophages that essentially are out of the circulation. The overall purpose of these cells is basically patrol that specific area. They could live there for years, right? And their job is to basically find any type of bacteria or foreign invaders that they could engulf, and then either phagocytize them or present it to lymphocytes in order to create antibodies, right, for them. So that's something we talked about here, right? Any questions about that, right? So
Yeah, I have a question.
Yeah, go ahead.
So did the cells get different names in the different tissues because they change in some way, or it's just because they're in a new location?
Yeah, usually it's location dependent, right? The overall name for them will be the histiocytes, right? That will be the overall terms. The common names you will hear is basically macrophages, right? The moment they leave the circulation, but specific tissues that definitely have the specific names. So some other cells, right? In the kidneys, I think they're called mesangial cells. So in the kidneys, they'll be called mesangial cells. So the same thing, right? And then I think in your skin, they're called Langerhans cells.
So it's not like they're getting transformed when they go to a new location.
No, they're still macrophages. They're not transformed, and basically, what their job is surveillance. Their job is to survey the environment and find any type of foreign bacteria and to engulf it and basically either phagocytize it or present it to lymphocytes to start building antibodies. Sometimes, though, in the autoimmune diseases, right, there is a problem where your body basically starts to take your normal body cells and think they are foreign. So we'll talk about some of those conditions. But for the most part, for healthy individuals, these are your regular macrophages. Their function is basically travel through the tissues or be in the tissues. And if there's a foreign antigen that's coming, or bacteria or pollen grain or whatever it may be, it's to basically engulf it and eat it up, chew it up, utilize its own lysosome internal granules, right, to basically break that particle down. Or if they cannot do that, they cannot kill it by just engulfing it, right, and phagocytizing it. Their next job is they're also going to become antigen presenting cells. We're going to talk extensively what this is. So basically, they'll take that antigen, they'll express it on the cell surface, right, and then they will take it to the lymphocytes. And then the lymphocytes they're going to decide what to do with it. We're going to talk about now about the lymphocytes. So I know Helmi was trying to steer me off course, saying monocytes are T and B cells. They are not. So lymphocytes are the T and B cells. Let me show you what they are. So let me do this. So I'll take the top portion of this. And I'm gonna add it here. Right? So let's talk about lymphocytes. So lymphocytes we can subdivide into a couple of different cells. Helmi, you want to help me here? I think you will probably be able to now answer, what the makeup of lymphocytes is.
E lymphocyte, B lymphocyte.
B lymphocyte, good. What else?
T lymphocyte.
T lymphocyte, very good. And one more.
I don't remember it.
Okay, so it's natural killer cells, right? So B lymphocyte, the T lymphocyte, and the natural killer cells. And not to make it too complicated, there's different T cells, right? We're going to keep it basic. So what I would say is there's different T cells, but basically what we want to focus for this lecture, we want to focus at T helper cells, right? And the T helper cells, what it does is it will basically signal, right? It will signal the B cells, the B lymphocytes, to basically transform into plasma cells, right? And the plasma cells is basically what they are, the activated B cells. And their whole job is to essentially make antibodies. They make antibodies against whatever the antigen that came in. So make antibodies, right? And antibodies could be, IgE, IgM, IgG, and so forth, right? So the whole distinction, right, of lymphocytes was basically we have the major ones is the B cells, the T cells, and the natural killer cells. And then the T cells, they further subdivide. There's T helper cells, there's T cytotoxic cells, right? T regulatory cells. For this lecture, it's enough to know that there's T helper cells that basically signal the B cells to transform into plasma cells, which is activated B cells, and their role is essentially to make antibodies. One important factor that I want to say is that natural killer cells, right, and your monocytes or macrophages, right, monocytes, your neutrophils, right, your basophils, and your eosinophils. These are agents of innate immunity. And B and T cells, they're the agents of adaptive immunity. So what do I mean, right, by innate and adaptive? So usually when, let's say, a foreign bacteria comes into your body, right, these cells are not the first to come and start to attack that. The first cells that come are these cells, right? The neutrophils, the monocytes or macrophages, as they're called in the tissues, basophils, eosinophils, and your natural killer cells. These would be the first lines of defense, I should say. And if the infection is ongoing, right, it's not basically taking care of these cells by the innate immunity, then we have adaptive immunity or specific immunity, right? That's basically is activated, which is made up of B cells and T cells, or B lymphocytes, T lymphocytes. And those are the cells that basically are the agents of adaptive immunity. And those are the cells that are basically going to come and then foster a response where they start to make specific antibodies focused against that foreign antigen or foreign bacteria or foreign virus, right, that comes in. So that's how we subdivide your white blood cells into these components. Let me make sure I have everything here, right? Yeah, I think I subdivided all of these there, right? So lymphocytes, right? Based on this chart, we said were the B lymphocytes, the T lymphocytes, and natural killer cells, right? The ones that I'm going to highlight in red, right? These are part of your adaptive immunity. So your body adapts to the antigen by making antibodies. And all of these that I'm going to go in green, right? This is going to be your innate immunity. And then the natural killer cells.